exactly 2:30. Now recording has started. You can start the presentation.
Let me start.
Ji, ma'am, you can start.
Thank you. Very good afternoon, everyone.
Yes, ma'am, you can start.
Yeah, very good afternoon, everyone. Myself, Dr. Santya, and I am working as a scientist in vegetable science in division of crop improvement at Indian Council of Agricultural Research Station, VPK's Almora, Uttarakhand. And I thank CITH for calling me for this lecture. And today I am asked to present a topic for you all in genetic enhancement for virus resistance in okra. As you all know that okra, we know it is an Abelmoschus esculentus, and its chromosome number is 130, and it is a polyploid crop. Being a polyploid crop, working in markers, especially due to presence of lot of intron region and lack of proper sequencing in this crop, working in molecular markers is highly difficult. Apart from that, in okra, DNA extraction is also difficult, little bit difficult compared to other crops because of its high presence of mucilage. These are the difficulties of this crop being a polyploid one. And this crop is mostly grown in tropics and warmer parts of the temperate region, and it is originated in Africa, that is especially Ethiopia, and from there it have spread to Middle East, and from then it came to India. And okra is very important in India. We can say that India is very important for okra because 72% of world production of okra is taking place in India only. That too in the production is around 6 million tons just in 0.5 million hectares area. We are producing. Am I audible?
Hello, ma'am. Dr. Ramesh, please mute your Dr. Ramesh.
Am I audible?
Yes, ma'am, you are audible.
Yes, ma'am, please continue.
Yeah.
Please,
So
Amita ma'am, please mute Dr. Ramesh.
fine. India is the country where 72% of world production is taking place. Next to this comes African countries, especially Nigeria. So next to okra is the breeding work of okra, and global okra conference usually takes place in India. And next comes Africa. Africa is the second important continent where okra is very important. And in case of India, it is grown in states like and in case of India, it is grown in states like Gujarat, West Bengal, Bihar, and Madhya Pradesh. And whereas in India, global export, in terms of global okra export, India contributes around 3.2% in terms of monetary benefits. And most of the Indian okra is exported to countries like United Kingdom and UAE, and European continents usually import okra much for their frozen products. Through okra, Indian export DCGS earns around 3 million US dollars every year. And not only in terms of vegetable purpose, in seed industry also very big boom in case of okra. Actually, next to tomato in terms of value, whereas in terms of quantity, okra seeds, hybrid seeds are produced largely in India. And in terms of value, tomato stands first, next comes okra for seed export. These are the significance in terms of monetary benefits. Apart from that, economic significance, fruits are mainly used for vegetable soup, sauces, pickles. Apart from that, industrial purpose, dehydrated, canned, and frozen products are also highly preferred. And okra mucilage, which is rich in polysaccharides, they are have use in packing industries, especially for packaging industries, for textiles, and also ice cream and bakery industries also it have use. And roots are used for sugarcane juice clarification, whereas stems are used as a fiber to form ropes, nets, mats, and fabrics, etc. Whereas there are also reports that the crop waste is used in bioenergy production and biodiesel production. So all parts of okra is significant in one or the other way. Apart from its economic significance, it have nutraceutical and pharmaceutical significance also. These okra pods are highly rich in iron, potassium, magnesium, and oil content also ranges from 35 to 50%. You all know that only few vegetables have iodine in them, and okra is one of the crop which is known for its rich in iodine content, and also it is highly rich in fiber. That's why every meal, na, they are preferred to have okra. Doctors are suggesting to have okra in their meal to increase the fiber content. It is having around 3.2 gram in it. And apart from this nutraceutical values, pharmaceutical values coming to that, it is rich in antioxidants, and also they have antibiotic, antibacterial, and gastro and immunomodulatory properties. And their polysaccharides is responsible for their immunomodulatory function, and it also helps to fight against cancer diseases. These are some of the pharmaceutical reports. Being this important, the major problem comes in okra breeding is, apart from shoot and fruit borer pest, the major problem is viral disease. Breeding for viral disease is highly difficult because of vast number of begoma viruses affecting it, and also large whitefly biotype evolution because of recent evaluation of large number of biotypes of whitefly and virus. According to them, breeding for virus resistance is highly difficult. It is reported that around 27 begomoviruses affect okra, and all these begomoviruses belong to the family Geminiviridae, as we all know. Hello, I am there. Am I audible? Is somebody? Okay. As I'm audible, somebody respond to me. You people are hearing? Yeah, fine. Most of these viruses belongs to begomovirus, and they are in Geminiviridae family. And apart from begomovirus, there are reports that some of the potyvirus also.
Yes, ma'am, please. Shoaib sir, you can unmute yourself.
It's everything fine. Let me continue. Am I audible? Yeah, fine. Okay, let me continue. And most of these viruses are single-stranded DNA viruses, and in India, among these 27 viruses, the most common disease which occurs is yellow vine mosaic disease and okra initiation leaf curl disease. And this yellow vine mosaic disease, you know, as you all know, symptoms like yellowing, twisting, stunting, and reduced fruit size occurs. Whereas in initiation leaf curl virus, there is the presence of pinhead-like structures and leaf curling. In North India, there are reports that both this yellow vine mosaic disease and initiation leaf curl disease both occur in the same plant, in same population. But in southern India, it have reported that once yellow vine mosaic disease comes, there is no incidence of initiation leaf curl virus, and there is vice versa. And this initiation leaf curl virus is caused due to monoparotid
Shoaib sir,
begomo.
can you say something?
Now we can speak. मैम एक्चुअली यहाँ पर प्रॉब्लम हो गई थी, मैम ने अनम्यूट किया था सबको मैम, इसीलिए हम नहीं बोल पा रहे थे. Now it's
You are unmute now.
you can go carry on, ma'am.
Okay, fine, thank you. Whereas Okra Abelmoschus disease is caused by begomaviruses, which are biparotite and monoparotite. That is, in case of biparotite, DNA alpha and beta satellites are present, whereas in monoparotite, there is only beta satellites present. In case of yellow wine mosaic disease, there are reports in India alone that more than 8 viruses are affecting this. Yellow wine mosaic disease is caused due to 8 different strains of viruses, among which Bendy yellow wine mosaic Delhi virus is biparotite. Apart from that, everything like Bhubaneswar, Haryana, Bangalore virus, and cotton leaf cull Allahabad virus and Okra leaf cull viruses are monoparotite in nature. And this Bendy yellow wine mosaic virus is having the resemblance, almost similar resemblance in sequence of DNA beta satellite to tomato leaf cull New Delhi virus. So these viral diseases are invariable of Bendy. It is affecting all the crops, like tomato leaf cull virus strains are found to cause yellow vine mosaic symptoms in Bendy. Similarly, Bendy yellow vine mosaic virus are reported to cause symptoms in tomato chili, and recently it have also reported to cause yellow vine mottle symptoms in cucurbits also. Recently, 2025, they have reported this. It is occurring in bitter gourd and pumpkin also. These are about different viruses. Being a viral disease, managing them is highly difficult. Both these viruses, that is common viral diseases, yellow vine mosaic disease and Venetian leaf curl disease, both these viral diseases are begomavirus disease, which is transmitted through white fly. So as we all know that viral management is a difficult thing, and only we can able to manage the vectors. So using insecticides, imidacloprid, acetamiprid, these are the normal sucking pest insecticides which is recommended to control white fly. And also neem-based sprays like plant oils and palmarosa for eco-friendly management. There are reports, but it is not commonly used that commonly. And there is one method, like once Bendy yellow vine mosaic virus and Venetian leaf cull virus, both these virus diseases is based on the stage of infection. In case of yellow vine mosaic virus, it is reported that it may cause yield loss up to 50 to 90%. That depends upon the stage of infection. If it is an early stage, then the crop loss will be more, as we all know. Whereas if it in later stage, 30 to 40% of the crops can be saved. So once if we found in the field some of the infected plants, it is advisable to rogue that plant and throw off from the field. And also it is recommended to use border crops to minimize the vector attraction towards the crop. And also Kharif season, especially the preferred climate is warm, humid climate, which is highly recommended for this white fly multiplications. To avoid this crop in that season can be done. We can grow in Zahi season, that is during March, April, we can grow this crop instead of Kharif season. But these are some of the suggested management. But the ultimate solution is not by managing to develop a resistant varieties. These are the advisable one, and there comes the role of not only cultivable varieties but also pre-breeding lines, which is having harboring resistance to various viral diseases. It is in current research demand of many of the breeders to develop pre-breeding lines harboring resistance that can easily incorporated into the breeding system. So as far as now, there are reports and there are development of many varieties using wild relatives for virus resistant. The developed one is Pusa Savani way back in 1950s by using local selection IC 1542 Pusa. This is a symptomless carrier. This IC 1542 is not reported to be highly resistant, but it is a symptomless carrier. So Pusa Savani became susceptible during that time itself. And after that, Punjab Agriculture University have developed varieties, which is Punjab Batmini and Punjab number 7 by using hybridization, where they have used Abelmoschus Manihot with esculentus cross. And later IHR developed these varieties, the resistance have broken down. And later IHR have developed Arcanamica and Arke Abe. These both are sister lines by interphosphation with tetraphilus. So for hybridization, have used these two wild relatives. One is Manihot and one is Tetraphilus. And Parbani also developed a variety by Parbani Pranti by using Manihot. So only these two species, Manihot and Tetraphilus, have been interspecific hybridization for genetic enhancement in okra. In okra, in wild relatives and land races are far more. These photos collected from NBBG at Thrissur station, where the variability of okra have depicted. And there are reports that apart from Manihot and Tetraphilus, there are reports that Abelmoschus moschatus and Abelmoschus caeli to be resistant to yellow in mosaic virus and okra Asian leaf curl virus. And you can see from the slide that the chromosome number of this wild relatives are highly variable. That is, it ranges from 72 in case of Ficunius, but Ficunius is reported to be susceptible to both the viral diseases, whereas in moschatus also there are reports that they have 72 chromosome number 130 due to their polyploidization, and during the evolution the chromosome number of the crop is not properly standardized. Not properly standardized in the sense it highly evolves. And so because of this varying chromosome number, hybridization, wild hybridization with cultivated okra faces, as cultivated okra chromosome number is 130, faces pre and post zygotic barriers. And once we use cultivated as a female parent, there are reports for high possibility. Whereas if they use as a pollen parent, and if you use wild species as a female parent, then the crossability is very difficult. And there are reports that embryo rescue have taken place to attempt a process in IRI. Apart from these are some of the photos depicting wide hybridization with cultivated esculentus and different wild species. So apart from wide hybridization, there are other methods also to enhance the genetic nature of the crop. And one among this gene editing technology, by editing the viral genome segments, especially replicating sites, we can develop genome edited okra lines. And also transgenic approach using RNA interference technology and BT Cry1AC genes. They have developed few lines, but this BT Cry1AC genes, they have developed 21 transgenic lines. This work have been carried out at Indian Institute of Vegetable Research, Varanasi, but this is for fruit and shoot borer resistant. Using RNA interference technology, they have used to suppress the viral replication sites, and code protein sites have been suppressed. And there are reports that RNAi technology have been used to develop a few transgenic lines, and that from Punjab Agricultural University, where they have used RNAi technology against AC1, AC2, and AC4 replication sites of virus, and they have silenced these genes to develop transgenic lines, which is showing low virus incidence. And also they have used RNAi intron hairpin specific RNA technology to develop virus resistant lines by attacking the beta satellites of these viruses. These two technologies have been developed by PAU and IAVR Varanasi. These two institutes are working in transgenic okra also currently. And this is a case study where the work in wide hybridization work, which is carried in Indian Agricultural Research Institute, New Delhi, where they have, as I said earlier, Manihot and Tetraphilus have been used in varietal development program. Currently, Indian Agricultural Research Institute is trying to incorporate virus resistance from moschatus through wide hybridization and development of variety. They have selected Abelmoschus moschatus as a source parent, donor parent, and Pusa A4 they have used as a reciprocal parent in this work. And they have attempted screening of this, and from their reports also it is proved that these are the source for both the virus diseases. The donor is highly resistant to both the virus diseases, whereas Pusa A4 showed susceptibility, highly susceptibility. So they have attempted F1 by both direct and reciprocal cross, but the success rate was high in direct cross, where Pusa A4 is used as a female parent. The hybridization have been confirmed using this 8 set of polymorphic markers between these two lines. Sorry, these two genotypes. They have confirmed the hybridity of the F1. But on going for pollination studies, pollen viability have started declining see, the viability of pollen is near to 100% when it is as a male and female parent. Once F1 is made, the F1 becomes sterile. The pollen viability of the flowers in the F1 have drastically reduced to 12 to 14%. So this have confirmed the F1 sterility of these two processes. And in this cross also, the attempt was good when Pusa A4 is used as a female parent. In that cross, that F1, they have again used as a female parent, and they have attempted the reciprocal cross, from which they could able to get around 24 F1s and BC1 F1 generation, by where they have undergone the inheritance studies, and the pollen viability of all these crops have been checked. And from the BZ1 F1 population, once virus screening and pollen viability have started increasing in BZ1 F1 populations compared to F1. F1 is highly sterile, and BZ1 F1 population, the sterility is declined and the pollen viability have started to grasp up. And they have underwent, the same group have underwent embryo rescue technique and also colchicular technique also. And after that, there's 24 F1 plants where BC1 F1 plants were screened, and most of them showed resistant to high resistance to these two viral diseases on disease characterization. So as cultivated, currently as the cultivated genotype doesn't have any source for resistance to these virus diseases, relying on wild relatives is the only way to develop resistant varieties. So currently IRI is in pipeline to develop resistant variety using Moschus as a source. And this is like how all the 24 F1s are similar to Moschus than the cultivated ones. So in this study, they have confirmed the resistant source of this genotype, that is Abelmoschus moschus. Through this pre-breeding study, they have identified this as a resistant source, and the pollen viability or the drastically reduced in F1, that is F1 sterility, also they have confirmed, and it started grasping up in BC1 and F1 population. And they have found out around 8 polymorphic markers between these two things. As I said earlier, okra is orphan crop in terms of molecular markers and omics breeding. And among 24 developed F1s, 20 were highly resistant, and these are in pipeline in the varietal development. This is a case study of work which is carried out in Indian Agricultural Research Institute. So apart from this wild hybridization and pre-breeding technologies, methods like RNA interference, marker-assisted selection, genome-wide selection, and CRISPR-Cas can offer to enhance the virus resistance. But in terms of okra, carrying out this is a highly tough job, as there is no any sequence. They have reported that recently in 2021, few segment of okra have been sequenced, but still that is not available in the public segment. So as far as now, pre-breeding is a good approach to develop interrogression lines having virus resistance, is a major objective of okra breeders. Thank you. Finally, I like to conclude my talk with a small thought that in all the things of nature, there is something of marvelous. So if we explore the nature's gift, then we will get all the results what we want. Thank you.
Thank you, ma'am. Thank you for this wonderful presentation and the wonderful work you have done with the okra and IARI. Any participants who wants to interact? Please, ma'am, unmute them. Unmute all the participants.
They are already muted, unmuted.
Anyone among the participants who wants to ask any question or has any query?
Excuse me, madam.
Yes, sir.
Yes.
What is the present